Thank you so much, Nicole. Thank you all for coming today. Lovely to see familiar faces and some new faces. Yeah, it's been really wonderful to be part of this group too. C-MAIKI, it's such a diverse group. And there's always a fun problem to work on from a mathematical perspective. This talk is more on my perspective of microbiome. Might be a little different from your perspective. Hopefully it's somewhat complementary. So let's dive in. So what is a microbial system for me? It's a highly dynamic system. It's changing. It can change rapidly in response to changes in the environment. And even if these microbes are dead, we know that just based on basic physics, they jiggle around like here in water, if they undergo Brownian motion. If there is some wind or water current, the dead cells can drift, move around. So this is known as a passive system. It's a passive dispersal. Now, what makes living systems and microbial systems interesting to me is that when the cells are alive, they can actively move. That's shown here in the bottom left. They can swim and actively disperse, and then you can get very different communities forming in different parts of the human body or the animals in different environments. So that's very exciting to me. Active system. So today I would want to present to you the mathematical physical viewpoint of in an effort to try and predict and understand this actively moving, this active motion and dispersal. Relatively well understood when these cells are moving in open space. So these are some mechanisms by which bacteria can move and disperse and occupy more favorable environments. This is one of the classical models of dispersal where a bacterium will will swim in some direction for some period of time. And then due to flagella, that they're the tail-like structures that rotate. And when the rotation reverses, it can do something called tumbling and randomly reorient. And after a series of these events, it can move much further than they would if they were dead. More recently, there's been some observations of more complicated behaviors like run, stop, run. So they stop, or they can reverse direction, run, reverse. So this is an open space. Recently, at least in the mathematical and physical community, there's growing interest in what happens when these cells encounter more viscous or tighter spaces that commonly arise in natural systems. Then there was an exciting discovery that flagella can wrap around the cell body in some in confined spaces, and they can undergo corkscrew-like motion. There's some surprising things that can potentially happen when they're in tight spaces. And tight spaces are relevant to many microbial systems because when we look in soil, for example, or in biofilms or in tissues, these bacteria don't have much room to move around. And in these environments, we know from physics that they move very differently from what they would do if they were had a lot of room. So this is an important observation that was made and studied about 20 years ago now. This is on a microscope slide, and they looked at bacteria swimming, and they found that bacteria that normally do run and tumble motion, they go straight normally, they tend to move in circles. And this was explained and studied based on hydrodynamic interactions. The flows generated by these bacteria get altered near solid boundaries. And this has profound implications. It means that they can't disperse as much as they would if they were swimming straight. And then more recently, the bacteria have been studied in porous media. They put some hydrogel beads, I believe, and saw that bacteria, they can remain trapped for some period of time before hopping to new sites. So this is very different kind of movement from what we were used to before. And then sometimes they can swim for long distances in a particular direction if you expose them to some flow. So that this can have important implications for infection. If the bacteria end up in places you don't expect them to end up. Here, the fluid is flowing in a certain direction, so you expect bacteria to also move in that direction. But in some situations, bacteria can migrate against the flow. There are some surprises that can occur in confined environments. And it's not well understood when you confine these bacteria. Where are they going to go? What's the direction of movement? How quickly are they going to disperse? These are generally open questions that for MAIKI are actively researching right now. So with this in the background, today I'd like to present two projects I've been involved with here in Hawaii that involve bacteria moving in confined environments. I'm dividing this talk into two parts. The first part is going to be about bacteria swimming in narrow passages. And then, so this was started thanks to C-MAIKI, actually. We started several years ago. And then more recently, we started looking at bacteria swimming on fungal highways. And yeah, in both these projects, where generally my personal interest is in understanding key factors or mechanisms that are governing the movement of bacteria. Where are they going to go? And then how quickly are they going to disperse? These are common questions that I'll be pursuing. Please feel free to interrupt at any stage. I welcome your questions. Are there any questions at this stage? No? Okay. In that case, let me start with the first part about bacteria in narrow passages. This is largely thanks to C-MAIKI, actually. We had all these people from different departments come together. We were all in Hawaii back then, and since then we've dispersed to different universities. The experiments were led by a postdoc at the time, John Lynch, and junior faculty Sang Woo. And with funding from C-MAIKI, we were able to perform some experiments. Okay, so let me try and motivate the project a little more. I learned from Margaret that there are a certain type of bacterium that recruited in the squid. There are a series of stages that help the squid to recruit this particular bacterium. One of the last stages is involves bacteria swimming through a very tight, narrow passage in the squid. I got curious because, as I tried to motivate earlier, bacteria can do strange things in confinement. So I got curious, how are they able to squeeze through these tight spaces? What's the bacterium doing before they eventually reach this light organ where the bacteria are needed or recruited? What's generally well known in the field is that chemical gradients can promote migration of bacteria. This is called chemotaxis. And I believe this is happening in the squid too. There are chemicals that are released in the light organ and that promote migration, that attract certain kinds of bacteria to this site. But what was not known at the time is what's the effect of this confinement on the movement of bacteria. So to study this, and I hope this cartoon shows the idea that it gets progressively more narrow as we get towards the light organ. We got curious, what's the effect of this gradual increase in confinement? So to study this, we created microfluidic channel. So this is not a real squid anymore. It's an artificial system consisting of a relatively open space on the left. It has a very high distance from the ceiling to the floor, But then there's a sudden transition to this more confined space. Here, there's only a gap of 2 microns or 10 microns. It's similar, comparable to the size of the bacterium. And here's a top view of what, let me show you a sample video of what we saw. So I hope you can see. I don't know if the contrast is good enough. Can you see some bacteria moving around? Oh, good. Okay, so as expected, maybe we do see bacteria going in and out. Yeah, there's room for bacteria to move into these confined spaces. What's surprising though is that here's the surprise. So we followed some of these trajectories, and what we saw is that when the channel is relatively tall, the trajectories are there's looks like spaghetti, but what you're seeing is tracks of bacteria. We're just tracking the position of each bacterium over time. We see a lot of tracks. Yeah, that's the idea here on the left. When we make the channels shorter in height, there's still room for bacteria to move in and out. But I hope you can see there are not many tracks here. This was surprising. There's still room for bacteria to go in there. And a few do go in. You can see some tracks. So they go in and then quickly leave. So this was surprising to me, to all of us. It's as though bacteria are afraid of tight spaces. Yes.
So for your narrow height, right?
Yes.
Does that mean that you're limiting the opening so fewer things can get in?
Fewer things can get in at any given time. That's true. The door is smaller, I guess, into this. But that is true. But just looking at the ones that do come in, suppose, yeah, what's surprising is that even when they come in, they quickly leave. Most of them leave quickly. Whereas the ones here that come in, they come in and stay in the room in this. I mean, some leave here too, but this was surprising to me. So maybe it's not surprising that there's roughly that not many are going in. Yeah, but it's the fact that they're leaving that was surprising. Yes,
What type of bacteria we use?
what kind of bacteria?
Yeah.
Oh, it's the Vibrio, yeah, was provided, yeah, Vibrio fischeri, yeah. Thank you, yeah. They have flagella and they can actively swim.
And is this without a chemoattractant?
That's right, there's no chemical gradient at this stage. We'll have chemical gradient later. And for now, we're just observing them.
How big is the bacteria?
Oh, yeah, thanks for asking. Yeah, so bacterium is it's about 2 micrometers in length. And and the diameter is small. I think it's on the order of a few hundred nanometers, or less than a micron.
Really squeezing through. Figure out one on the right.
On the right, yeah, well, I like to think that the diameter is less than a micron, so there's still room for the bacterium to winkle around. But the length, to include the length of the flagellum, it could be longer than 2 microns. But yeah. Yes.
Did you only test 10 and 2?
We may have had a few other ones too. Yeah, 5 microns, but we saw.
Between.
Yeah, it was done several years ago. I'm trying to remember what height we used. I wanted to make it even smaller than 2 micrometers, but the collaborator said, yeah, it does get harder to make these channels that are narrower. Yes.
I'm sorry if you already mentioned it, but are these like quasi two-dimensional channels, or do they also have some height to them? Like, so we see the plane here.
Yes.
How deep, I guess, are these channels? So it's 2 micrometers across this way, but if it were to come out,
Oh, you're asking about these dimensions? Yeah, so the width is about 50 micrometers, and then the height is like
Okay,
two microns in yeah, or 10 microns.
so they do have some room to kind of, in the 3 dimensions to turn around.
Yes, they have plenty of room to turn around. Yeah. Yeah, 50 microns is quite wide. Yeah, so yeah, thanks for asking. Yeah, Okay, so these are just qualitative observations for now. We wanted to quantify what's going on, what the difference is between these two cases. So here's some. We looked at quantities like the swimming speed, but there was no noticeable change in the two cases. They swim at roughly the same speed. What is different though are the directional changes. What I mean by that, I look at how the direction the bacterium is swimming in, and I look at how much the heading angle changes over time. If we take the square of the magnitude of the change, the directional angle change, and plot how it changes with time, it typically grows, that they tend to randomize over time, but it changes more quickly in the 10 micron channel than the 2 micron channel. This means that the paths tend to be straighter under confinement. This was one important quantitative difference we saw. We can explain this because in terms of how easy it is to turn. When the cells are more confined, it becomes harder for the cells to turn, even though there's room to turn. In order for the cells to turn or change direction, the fluid has to flow in the right way, and there's more additional resistance when you have a very thin gap of fluid between the cells and the surrounding walls. So that's one important difference. The other difference we saw is that I should have mentioned, these cells, they run for some time and then they reverse direction after randomly. And if we look at the duration between successive reversals, that typically has this exponential distribution that's shown for the yellow bar in the 10 micron channel. But we saw that it becomes different in confinement. In 2 micron channels, we saw very few short runs. So it seems like these reversals get delayed in more confined, under confined, for reasons we still don't really understand. So these are the quantitative differences we saw. Yes, Jörg?
Can you look at the tumbles to see, like if you're in the confined space, if you're tumbling in the Z direction, does that occur less than if you tumble in the white plane, like the 50 micrometer direction? I mean, it's probably hard to see, right? You can't determine how they tumble. I
when
could
you say
see
tumble,
the vertical tumble being impaired, but the
Yeah.
horizontal tumble.
Yeah, that's what I'm plotting here. Where it's essentially it's not moving much in the vertical direction, but it is turning in the horizontal in the. That's
No,
what
but
it's
the
called.
tumble, when it tumbles, it goes in all.
In all directions, yeah, but in the 2 micron channel, there really is
Right.
not much room for the cell to
Yeah.
go up or down, and so it's mainly rotating like this,
Right.
and that is the directional changes are reduced.
Yep.
Yeah. So based on these observations, we created a model. So since I'm a mathematician, I thought I should have at least one slide with an equation. Here it is. I think this is the only slide with an equation. So this, based on these observations, we create a model. We think a simplified version of what we think is happening. Here I'm predicting the change in the distance into the channel. That's given by this term here. I'm saying it changes because it's bacteria swimming at some speed u, at some angle theta, and it changes over time, delta t. And then this equation says the direction angle theta changes randomly. This is the magnitude of the change. And then to this we also add a reversal. So occasionally it will reverse direction. And just based on these equations, we can simulate some facts, and that's shown here. Let me start with the simulation. So in the more confined case, in the new 2 micrometer channel, I'm plotting how facts for bacteria going into the channel. And the red curve shows the average of 100 bacteria that are simulated. And you can see most of the bacteria leave the channel. Yeah, very few remain after 10 seconds. Yeah. This is consistent with what we saw in the experiments. I'm comparing here with the bottom left panel. These are experimental data showing the bacteria going into the channel. Very few remain, and the red curve also goes down to 0. In contrast, in the less confined channel with 10 microns, yes, some bacteria do leave, but about half of them stay in the channel. And the red curve doesn't actually drop down to 0. This is consistent again with what we saw in the experiments. So we think that a combination of reduced directional changes and delayed reversals can explain why most bacteria ended up leaving the channel. The picture I want to paint is that if the paths tend to be straighter and you reverse, then you're more likely to go back where you came in from. So bacteria tend to leave. Whereas here in the less confined case, the direction is changing randomly. So after reversal, it may not go back to where it came in from. It can stay and linger in the channel for longer. So what you might be wondering? Well, this has some broader implications. So some of you asked, there was a question about chemical gradients. We know chemical gradients are important in microbial systems. Here we created we looked at a tapered channel, and now we created a chemical gradient promoting migration from right to left. And as expected, when the tip is quite large, we saw bacteria migrating to the left and leaving this tapered channel. The white parts represent the parts with a lot of bacteria, showing abundance of bacteria. What was surprising was that when the tip of this tube is very narrow, it's only 1 micrometer, we saw a depleted zone with not many bacteria. So over time, most bacteria end up in this intermediate, in this middle zone of the tube. And we think it's because there are two competing effects. There's chemical gradient promoting migration to the left, and then there's this competing effect I tried to paint earlier, that there's due to confinement. And there's strong confinement, bacteria tend to escape from this region. So you get this non-trivial distribution of bacteria. So the take-home message from the first part is that not just chemical gradients, but physical gradients can potentially impact the distribution of bacteria and where they end up in our system. Yes, Masara?
So my understanding then, so if I'm wrong, please correct me. So the confinement of the depth, like 2 micron and 10 micron, itself is going to influence the turning frequency of the bacteria, which is reflected in your model.
Yes.
So my question is then that if so, that's turning frequency is probably depending on the bacteria species.
Yeah. Absolutely, yeah.
Yeah, and the Vibrio is in this system, it's very filtered out. Only the few of the individual, just a random chance they can get in. But the other bacteria, they might have a much different parameters, so that
That's true.
we cannot get into the endpoint.
Yeah,
That's what
yeah.
you think it can.
Yeah, what I presented was just for one species of bacteria.
Right.
We put different bacteria in there, may see different responses to this. But yeah. Yes.
I'm sorry if I missed it, but do you know the size of the tubules in the light organ? Like
Oh, the
how,
tubules. What are the tubules?
yeah, like so for Vibrio fischeri, when they go into like the light organ of
Yeah.
the crypts, they're squeezed through. What's the size of that, like for that bottleneck?
Yeah, I asked my collaborators that they tell me that in reality the light organ is more complicated because there are I believe there are cilia growing on the
Yeah.
walls and some parts look. I've seen pictures that have about 2 micrometer gap, but they can be maybe 5 microns. It's more complicated than the microfluidic channel we looked at in our experiment. Yeah. believe these cilia are active too, so that they're creating flows that are neglected in. Yes. question, but-
I'm curious about the procedure. So do you allow the serine to be well mixed in the system first and then you introduce the Vibrio? Or do you allow the Vibrio to be well mixed and then you can put it serine?
Oh, how was this experiment initiated? Yeah, I believe. I'm trying to remember how we did this. So, yeah, excellent question. Yeah, I believe we made sure bacteria, it was several years ago when we did the experiments. I'm trying to remember how it was set up. I believe I remember seeing bacteria that were mostly at this end of the tube. And then, and we had a deep pool of serine outside the tube.
Diffusion,
Yeah,
just diffusing into the tube.
yeah, that's right. And then initially this tube did not have any serine. But when we put this tapered capillary tube in the pool, serine was able to diffuse slowly into this tube. That created this linear, the gradient. Yeah.
Kind of following up on that, why serine?
Why serine? Why not? We just wanted to see chemotaxis. I was told that serine is a known chemotaxin of this particular bacterial species. So use serine. Yeah. Yeah, try.
So when they behave differently than different confinements, what is it they're actually responding to? They're like, is it pressure or how do they know it's?
I don't know. Yeah. So I believe one possible response is that they even if they're just mindlessly swimming by rotating their flagella, just the presence of a wall nearby alters the fluid flow generated by the bacteria in such a way that they don't turn as much, or they're so that alone can impact where they are oriented and where they end up.
You think the bacteria are doing it themselves, like mechanosensation or?
That's an additional factor, yeah, it's not needed, but if there are additional responses, like if the bacterium is sensing something different in the environment and changing their activities within the cell, that can additionally lead to different responses, but I believe that is not required. Bacteria can continue doing its own thing, and it will respond differently depending on what's out nearby. If there's a wall nearby, yeah, it changes.
1 micrometer is really tiny for these bacteria, right? So you
Yeah.
almost have to come in with a straight shot, right? If you go a little bit off,
Yeah.
you hit the glass. And I don't know if they would stick for a while before they, sometimes when I look in a microscope, bacteria stick a little bit when they encounter a surface, yeah.
Yeah, what you're thinking about this 1 micrometer
Yeah,
here?
it's really tiny,
It
right?
is really small, that's true, yeah,
Cool.
but I'm told that in tissues or biofilms that there are lots of places where, yeah, the bacteria just don't have much room to move around, yeah, so. It was designed to mimic this kind of situation. Yeah. How are we doing with time? I think we're about halfway. We're about halfway. I wanted to take a halftime break, give ourselves breather and allow you to ask questions. You already asking plenty of questions. Wait, is there any other questions related to the first part, unrelated to the first part? I'm open to chat about anything else. Today, how's semester going for everybody? About halfway.
Were you guys able to do any follow-up physiology of the ones that made it all the way down the very narrow channel? Like, have they been selected for a longer flagella or?
Oh, that would be a really fun study, yeah, to see if there's anything unusual about cells. We did see some cells that end up in these really confined spaces, and yeah, that's an excellent question. We're not sure. Yeah, we didn't follow up on.
What's the orientation of flagella in the bacteria you guys use, Vibrio, right? What's the, it's just a single polar flagella. This- Or bank.
It's a polar flagellum and you might have up to 3 or something. It's not very.
Yeah, there's a small number of.
It's
flagella.
1, 2, or 3. And it's a sheath flagellum,
Yes,
which
that's
is
right.
different. So the outer membrane stretches over it.
I wonder if you added to our bacteria, the different setup. What it would look like.
Yeah, that would be fun to study, yeah. But different bacteria, yeah. Okay, well, yes.
Do they behave differently alone versus together?
Not so sure, actually, we, in all experiments we looked at, there weren't that many bacteria, so they were moving independently in isolation. And I believe that's also in the case in the squid, there's a very small number of bacteria. I think only one is needed to, for the cascade of events to occur afterwards. So the natural habitat, I think, consists of very small dilute suspension, small number of vibrios. But you're thinking about like a large population of bacteria swarming, yeah. Yeah, so that's more maybe for the second part of the talk, so maybe we can go move. Are you all ready for the second half? Yeah. Oh, maybe one more question, yeah.
Do you know how deep the biofilms went deep into the channel? So I noticed in the last slide that the second channel had buildup of bacteria on the walls. Do you know how far?
Oh, like here?
Yes.
Oh, interesting. Yeah, good observation. Yeah, you're right. Yeah. Yeah, I think these bacteria commonly accumulate on surfaces.
Yeah.
Yeah. So what is the question?
Oh, like, do you know how deep?
How deep?
Like, I guess, do you notice like at some point, or sorry, the second channel, how deep the biofilms ended in that channel?
Oh, you think there's a biofilm
Or
developing on?
if there is.
I'm not sure if they are if they've had time to build biofilm here, because remember they were initiated without bacteria. So this is maybe within a few minutes.
The dots are tracks, right? They're not stationary.
That's right, they're all constantly moving around, yeah. They may not be biofilm, yeah.
Wow.
Yeah. Okay, I'd like to move on to the second part of my talk, which is my more recent work that started thanks to collaborator New here. And a lot of the work that I'm presenting was done by the undergraduates. So William Gao did a lot of analysis, and the experiments were performed with Luke and Guy from here, New's group. So yeah, I'm very excited to show this preliminary work that that's ongoing. We got some support from the Europe program for the undergrad research, and then recently, as Nicole mentioned, we have gotten a grant from the joint program supported by NIH and NSF that promotes research at the interface of mathematical and biological sciences. Yeah, again, let me try and motivate what I'm about to present. So now looking at fungal mycelium, which is a network of long, slender structures called hyphae, and they can penetrate into tight spaces like soil. So here, this is a my understanding of what's happened in the soil. There are plant roots that are shown here that need nutrients. But unfortunately, when the soil is not saturated water, when there's not much water, water ends up in these little pockets that are isolated. And nutrients are hard to get for the plants. However, the fungal mycelium can connect these seemingly disjoint patches of water by growing further out. And interestingly, I was very excited to learn that bacteria can move around these places. So these, again, very tight spaces. And here's another paper showing that the mycelium or the hypha can be coated with a film of water, and there's been observations of bacteria moving in these films. So again, very tight spaces, and bacteria are moving actively in these spaces. So that's interesting. They can again, it's not well understood where these bacteria are going, and how quickly they're spreading in these spaces. So that's what I'd like to present today. Here's an overview of the experimental setup. We have a Petri dish with agar, and the fungus and bacteria are grown on one side, and there's a trench that's created for us to see under a microscope what's going on. So here's a typical video of what we see under the microscope. These long, slender things are the hypha, and you can see a lot of bacteria near these hyphae. There must be some water around them. This is quite complicated. There's a lot that we don't know about yet. Let me show you another video that with a smaller number of bacteria. Again, this is the hypha. Are you able to see the bacteria moving? It's very exciting to me. Bacteria moving in confined spaces. You don't really know what's going on. If you look carefully, it looks like they like to travel along these filaments. There's another hypha here down here. Something interesting seems to be happening near the tip. This is a tip of a hypha. What I'd like to focus on is the direction of movement. They seem to be moving along these linear structures. We replaced the hypha with synthetic fiber, with the fiberglass, and we saw the same thing. We have film of water around the fiberglass. This is the snapshots in time. You see these the bacteria. It's as though they like to move along these structures, like traffic on roads on a freeway. So these are qualitative observations that we made. Initially, we wanted to quantify whether they really are lining up or along these structures. So but before, this was very surprising to me. I want to emphasize how surprising it is, because remember, bacteria are jiggling around. Their direction of motion can change randomly quite easily, so that it's very hard for them to move continue moving in a particular direction. That's not observed in open space. But here it looks like they're able to move in a particular direction. So to quantify this, we looked at the orientation of the bacterial cells. The cells are elongated, so we were able to track the endpoints with and connect with the line. Now I'm calling the angle that this line makes with the direction of hypha by angle alpha, and that's shown here. We've bin them into these 6 ranges. So small angle alpha means bacteria are almost parallel to the hypha. If it's close to 90 degrees, it means the cells are oriented perpendicular to the hypha. What you see is that in the controls, I'm not showing today, but put them in a deep pool of water, we see almost a uniform distribution. That means cells are oriented randomly in no preferred direction. However, when we look at bacteria in near the film around the fiberglass, or in Haifa, we see a very different distribution. Most about half, a lot of the cells are oriented parallel, almost parallel to the linear structure. That was one quantitative result we got. That's just the orientation. If we now look at the direction of movement, so here I'm taking two successive frames. So this is the location of the center of the bacterium in the previous frame. And I look at where the bacteria move next frame and connect with the line again. That line will create an angle theta with the hypal direction. I'm calling this the displacement angle theta. That's plotted here. This is the distribution. Again, in a deep pool of water, we see almost uniform distribution for the direction of movement. But in a thin film of water around the fiberglass and Haifa, again, we see a similar distribution. Most cells are moving parallel to the linear structure. This is exciting. This is curious. I want to emphasize there is plenty of room actually for the bacteria to turn. The water film, we believe, is on your tens of micrometers. That's much longer than the size of the bacteria. So bacteria could turn, but somehow they seem to like to align with the Haifa or fiberglass. Now I want to show what's going on over a longer time. So here, maybe in the interest of time, maybe I'll just highlight the main thing. So here I'm just looking at the correlation, how much the angle theta, the direction angle changes with time over longer time. Typically, when you look at the cosine of this angle, it starts at 1 but decays to 0 over time because bacteria change direction randomly. So this angle theta on average will be it can be anything, and so when you take the average, this becomes close to 0. And here I'm looking at how quickly it decays to 0. This gives an indication of what's known as persistence time, how long bacteria tend to move in the same direction. And if we look at this in a deep pool of water, it is a pretty rapid decay. You have to wait only about 1 second or so, and then the bacteria will no longer be moving in the same direction. In contrast, in a thin film of water around fiberglass and Haifa, these curves decay more slowly. You have to wait maybe 5 times 10 seconds for the bacteria to change direction. And they do eventually change direction. They can reverse direction. And we see this, but it takes a much longer time. You have to wait 5 or 10 seconds for bacteria to change direction. Okay, so you might be wondering, so what? Well, this has very broad, very important implication on how quickly they disperse over time. Here the way physicists like to quantify this is to look at the square of the displacement. This is how much distance bacteria has traveled after a certain amount of time. And it's good to plot these on logarithmic scales because at early times, for very short intervals of time, bacteria are swimming. So we expect the distance to increase, the distance squared to increase like time squared. And that time squared means on logarithmic scale that these data points have a slope of 2. But if you wait long enough, after a long time, like the area occupied by the bacteria, this becomes more diffusive, and the area tends to grow linearly with time. And from these, then it gets more complicated later, but I just want to emphasize the red data points. This has a slope of 1, and from these data, we can extract how quickly they diffuse, they effectively diffuse, and from this we can measure the dispersal rate, how quickly they disperse. So here I'm just showing the data in a deep pool of water in Haifa and near fiberglass. When you're Haifa, we notice that bacteria tend to swim more slowly. That's why the area occupied by the bacteria tends to be a little smaller after a given time. So this is what we do with to quantify the rate of dispersal. What's surprising is that now when we look at the same thing now on near in a thin film of water around fiberglass or hypha. Again, now I'm looking at the where the displacement along the hypha or fiberglass. Again, we see this trends, but I want to emphasize that for firstly, the transition to this diffusive behavior occurs at a later time. And this is still ongoing work. I have but the take-home message from this is that these bacteria seem to be diffusing more quickly, they're dispersing more quickly along these filaments. And there is some mathematical modeling that can help explain this. We think that when they're changing direction, when they take a longer time to change direction, we can expect them to disperse more quickly. This has broader implications. It means that if it happens, even if the soil is not, or the environment is not saturated with water, these hypha can create passage for bacteria to diffuse very quickly. Yes.
Do you think that the water actually moves along the hyphae, like it's not static, so there could be some motion due to like, it's water tension kind of like?
Possibly, yeah, that may happen. In these experiments we've done so far, we're seeing bacteria moving both directions, so we think there's minimal background flow of water.
So I was thinking if you had beads, what does a fluorescent bead do, or what
That's
does
an
a
excellent-
non-motile or non-chemotactic mutant do?
Yeah, that's an excellent question.
You want to legal advance.
They wanted that.
That's one of our aims.
Yeah, that's for future work. Yeah, we'd like to study what the fluid is doing. Yeah. So. Okay, so I think we're almost out of time, so I should maybe start wrapping up. So in conclusion, I hope I was able to convince you that confinement can have surprising effects on the movement of bacteria. In the first part, we saw that tight spaces can cause bacteria to escape from them. And in the second part, we saw that when you have a linear structure with water around it, bacteria can keep swimming for long distances, for a long period of time in the same direction. Something we would not see in a deep pool of water. And so this impacts how quickly bacteria disperse over time, where they're going to go, and this can potentially alter the microbial community structures, depending on the traits of bacteria. And yeah, so what I presented in the second part is the early stages. I wanted to end with an advert for a postdoc position. The grant is going to support. This is for 3 years, and we're looking for new postdoc to join us. If you're interested, or if you know somebody who might be interested, please spread the news. We're looking for somebody with experience who can do both experiments and some modeling. And yeah, the qualified selected candidate will be working with both our groups and learn about interdisciplinary research, how to combine math and biology. Yeah, please spread the news, or please consider applying if you're interested. You can email me for further information. Okay, thank you, Nicole.